Okay then, welcome back to another episode of Brain Biology and Beyond. And today we're talking about the use of psychedelics in medicine. And we are going to cover the following objectives: to understand and revisit the monoamine hypothesis of depression, something that we spoke about in a previous episode on depression. See the link in the description. Appreciate some of the flaws with the monoamine hypothesis, and also to then discuss the use of psilocybin and ketamine as potential therapeutic agents, to evaluate the uses of them as well and potential barriers to their use in the treatment of depression and other mental illnesses. Before I start, this is not a medical advice for depression. If you are in crisis, then you need to make sure that you speak to GP, or if unable to, then you call Samaritans on 116 123. If you're in school, then please make sure you contact the pastoral team and member of staff. So, what causes depression? So, very highly contested within the literature. Traditionally, scientists have argued that chemicals cause this, with low levels of serotonin being attributed to causing depression. However, a lot of research conflicts this claim or suggests that the view itself is overcomplicated. Now, traditional antidepressants work by sort of stopping the reuptake of serotonin back into the synapse that you can see here at the top, by blocking the pump that takes it back in, and this leads to there being more serotonin present within the synapse, and therefore more serotonin able to bind to the receptors and do its thing. Now, serotonin itself likely has a small role within mood and mediates depression. However, the levels of serotonin go up immediately after taking these drugs within a few hours, yet they don't become clinically effective until 4 to 6 weeks after continuously taking them. Now, why that might be, to cut a long story short, would be because they actually work through downstream pathways which increase the expression of certain genes, such as BDNF, which are involved in neuroplasticity, which we're going to talk a little bit about now. Okay, so that's the main issues with antidepressants there I've just spoke about. And there are many criticisms of the serotonin hypothesis based on the fact that they have shown little benefit compared to placebo, i.e. pills that only contain sugar. Only about 33% of people respond to them, which is equivalent to the placebo effect. And serotonin levels obviously rise very quickly after taking them, yet the benefits don't seem to occur until later. So neuroplasticity, which I just mentioned, is the ability of the brain to form new connections and get rid of connections throughout its life. Okay, now we are most plastic in our brains when we are developing children and young people. However, there is the ability to tap into this plasticity throughout life, through various activities which enhance plasticity, such as exercise and meditation, as well as the different medications that we've spoken about. And what we're going to speak about today, the psychedelic medications which can alter neuroplasticity. Now, neuroplasticity can be good, it can be bad. Okay, it is associated with repeated behaviours and is very important within addictions and habit formation. There are neuroplastic changes during learning and memory, which is why continued revision would be important, and musical training appears to increase the size of various brain regions. Now, these are just a few examples of plasticity in action. I don't want us to think that plasticity is just a good thing. You can have bad plasticity, particularly in the cases of addiction, where bad habits can form and become more automatic as a result of the addiction. Plasticity is merely the ability of the brain to rewire itself. I'll speak more about regions of the brain that are important within depression in the episode I did on depression, but often areas that are important in regulating mood, such as the prefrontal cortex, become underactivated, and other regions such as the amygdala, which are involved in fear and anxiety, become overactivated in depressed patients. So activating pathways which can rewire and help restore that plasticity to where it should be is important. Plasticity in itself is just a remodeling. It's where that plasticity that happens and the context it happens in, which is really important. And that will come into play when we talk more about how these different drugs that we're going to talk about today work. So here are 3 examples of illicit drugs on the screen: ketamine, psilocybin, and LSD. And all of them are illicit and illegal and have the properties that they induce psychedelic effects. Now we're going to talk about ketamine first as a potential exciting antidepressant, which has been shown to have a response rate of up to 53% after a single dose within some clinical trials. It works by blocking the NMDA receptors and increases the level of a protein called AMPA, which leads to the neurogenesis and the formation of new neurons, which is important because depressed people tend to have lower levels of neurogenesis compared to healthy controls. Ketamine seems to have a long-term response after a single dose, with 71% of people reporting benefits after 18 days of that single dose. And unlike other antidepressants, it acts quickly. In fact, it can act more or less immediately, and this is due to the fact that it directly binds to the NMDA receptor. Now, that NMDA receptor is involved within the plasticity process. Antidepressants seem to take about 4 to 6 weeks to be able to activate that NMDA receptor and lead to, plasticity proteins becoming activated, whereas ketamine seems to be able to do it very, very quickly, and which was what leads to its rapidly acting effects, which is where most of the promise for it comes from. It has been approved within America under supervision, okay, and all of these trials that I've mentioned have been carried out under supervision. They are not using ketamine from the street, they're using pharmaceutical grade pure ketamine under supervision with experts to be able to help people should any side effects occur. Now, perhaps more interesting is psilocybin. Now, psilocybin has been used for thousands of years by people. It was only actually found to be a psychedelic drug within the 1950s. It is naturally occurring in over 150 species of mushrooms and produces hallucinogenic effects known as a trip in people that use it. And like other psychedelic drugs, it was outlawed in the 1950s and 60s. Partly due to the dangers, but also partly due to the fact it was part of the counterculture movement, the hippie movement, and there was some fear from governments that it would lead to people not working properly and potentially rebelling against the status quo. I'm not going to talk about the politics today of this, but that is something that you can read about if you're interested in reading about it. A good book would be How to Change Your Mind by Michael Pollan, where he talks a lot about the previous work that was done prior to psilocybin being banned. Interestingly, now that research, has been revisited with a growing interest in new antidepressants, and the fact that psilocybin, as we will come to see, has got a relatively low side effect and is relatively safe when we look at it. So how does psilocybin actually work? If I go back to that picture there, this is the shape of psilocybin, and psilocybin has got a very similar shape to another chemical important in the brain called serotonin, which we've spoke about a lot. And so psilocybin, what it does, it will bind to the serotonin receptor due to its similar shape. And when it does so, it activates various G proteins within the cell membrane, which is shown on this diagram here. That G protein activates another protein called phospholipase C. Phospholipase C leads to increased levels of inositol-free phosphate and calcium levels. Now, these things may seem insignificant, but they're just increasing the levels of these signaling molecules within the cell, which are then going to activate a lot of different cellular responses within the cell to cause something to happen. And as a result of the inositol-free phosphate and calcium levels reading, we end up with protein kinase C, which activates various proteins, including the BDNF and the AMPA, which are key proteins involved in neuroplasticity. So this is happening down a bit of a cascade of events which happen within the cell when psilocybin binds to the serotonin receptor. And you can see again that there is, at the output, there is neuroplasticity proteins which are being activated, and they can become activated rather quickly as a result of taking psilocybin. And it is right in thinking that taking psilocybin can permanently change people's brain structure. And indeed, it can have effects on personalities, which, some people might argue is good, some people might argue is bad. I'm talking about the context of it being used within depression, in medical settings and clinical trials. This is not to advocate using it recreationally. So there has been a number of promising studies looking at psilocybin and how it is used as an effective antidepressant, with reductions in depression after a single dose being reported for as far as 6 months by one study in 2016. Similar results have been found in those with end of life care, allowing them to come to terms with their illnesses and feel more at peace. There is an extremely high lethal dose, approximately 3,600, which is 40 times safer than citalopram, which is one of the leading antidepressants. Some long-term studies show that it has got a reduce in depression up to 2 years after taking, up to a year after taking 2 doses, which shows great promise there. And the potential mechanism of action includes altering brain network connectivity, including neurogenesis, so enhancing neuroplasticity within various regions, which I'll talk about in a minute. Most studies in this area seem to have come from private companies, although many governments are starting to fund research again after a long time absence and the growing need for treating of depression. Now, the way that these trials would have worked and has been found to help people through depression is not just by giving the people the drug on its own and just letting them be. That would be irresponsible and dangerous, particularly in the case of psilocybin. When it comes to psilocybin, we have people taking psilocybin at the low doses under the supervision of an expert. And that expert is going to help guide them through their trip or through their psychedelic experience and their mythical experience. And that mythical experience that people experience while under the influence of psilocybin has been found to be key for the therapeutic benefits to be ascertained. And indeed, if people don't have such high levels of the mystical experience that they have while under the influence, then it's potentially that the benefits are not going to be as great. So like other medications, you can't just give someone the chemical and expect them to get better, as is the case in typical antidepressants. They have to have some sort of guidance through an experience, often from the reports where they end up confronting something perhaps that they have been subconsciously repressing for a long period of time. So some of the potential mechanisms biologically for this are the increases of neurogenesis, with mouse models showing a 12% increase in dendritic spines, which are the connections on the ends of the neurons, and also greater global network communication. So you can see there in the picture that on the left you've got someone who's not taking psilocybin, and on the right you can see the networks of the brain communicating with each other much greater while under the influence of psilocybin. And that can, be a temporary state. However, some of those networks are going to be, more greater connected afterwards, or lesser connected, as the case may be. And one of the interesting networks and regions that is altered after taking the psilocybin is the prefrontal cortex and the default mode network. And there seems to be an enhancement of the prefrontal cortex following psilocybin and its ability to control fear responses. And also there seems to be a quietening of the default mode network, or the DMN. Now, the default mode network is involved heavily within thoughts of the self, autobiographic memories, and it is necessary for our concept of self. However, in people with depression, they tend to have a hyper-regulated or a hyperactive default mode network where they excessively think about themselves and to the cost of everything else. Their worldview is narrowed. People who take psilocybin tend to have a quieter default mode network afterwards, and this may lead to reduced rumination and therefore reduced depressive symptoms. And that is one of the leading hypotheses. Have a little think about that.